Hey, welcome back to my channel. Today we are checking about glycogen metabolism. Currently we are discussing about glycogenesis and glycogenesis. Glycogenesis, first we have known that the glucose is stored in the animal stored in as glycogen, plants is stored as starch, and 6 to 8 percentage of glycogen stored in liver, and 1 to 2% are stored in muscles. And the muscles have because of heavier muscles, they are 2 to 3 times more than glycogen in muscles than liver. And the main function of glycogen to maintain blood glucose level. Where during the meals, the glycogen is stored as in well freed state. When we are taking fasting, in that day, this stored glycogen is used for producing ATP for other purposes. And the glycogen is a homopolysaccharide with alpha D glucose residue. And the glycogen will have contain thousands of glucose residue, and with are branched with 1,4 glycosidic bond. And when the 8 to 10 glucose residue, then it is became branching with alpha 1,6 glycosidic linkage. Got that? And this glycogen where is it stored? This stored in systole. This stored in systole. It is stored in small granules form. Then we have it needed of glycogen. It is take. This we are going for glycogenesis and the glycogen. And the first step is glycogen is converted glucose 6 phosphate. Glucose is converted into glucose 6 phosphate. And in the muscles, this enzyme is formed exokinase. Here we are discussing about liver. The liver, it is glucose is converted glucose 6 phosphate by glucokinase, and with the help of ATP into ADP. And the glucose 6 phosphate is converted into glucose 1 phosphate by the help of the enzyme is phosphoglucomutase. And the phosphoglucomutase convert glucose 1 phosphate, and this is a crucial step. And glucose 1 phosphate is converted into uridine diphosphate glucose, and the UDP. And the UDP is glucose is converted by the pyrophosphorylase enzyme, and that converted UDP into UTP. And the later the UTP is formed, then the glycogen initiates synthesis, and then we have a primer, to synthesize. Glucose, and this primer, they are naturally the glycogen primer. Glycogen is acted as a glycogen primer, and that is in the nature is available. But the absence of a primer, a protein called glycogenin will present, and that acts as a primer. First, glycogen is initiator, and this glycogen initiator introduce one glucose residue to glycogenin, and then after that glycogen itself added other glucose residues. And after the glycogen primer, then the glycogen synthesis. Add the glycogen residue into 8 to 9 residues. Then the glycosyl 4,6 transferase. The glycogen is 4,6 transferase. It turned to branching. This enzyme is responsible for branching that form alpha 1,6 bond. Then it is elongation Then this bond is became elongation straight chain with glycogen synthesis and forming one for linkage. Then became branching. Then became branching with the glycosylase 4,6 transferase and then become 1,6 bond. Then the transfer branch. Then they became elongated branching, elongated, but it is a continuous process that became finally it formed glycogen. And here this is the diagram. This is I showed in this is the diagram, and before we have to know this is the main four steps. First step is synthesis of UDP. Second is synthesis of primer and primer of initiation of synthesis, and third is glycogen synthesis from glycogen synthase, and fourth is the last reaction probably, and that is the forming of branching, that a branching take place. And first the UDP, initial UDP, that a glucose converted into glucose 6-phosphate with the help of glucokinase, and glucose 6-phosphate is converted into glucose 1-phosphate with the glucophosphamutase and then the glucose 1 is converted into uridine diphosphate. That time the uridine diphosphate is formed by the UDP and phosphorylase enzyme. Then because the UDP glucose is formed, then glucose is formed, and then is a synthesis of primer. Synthesis of primer is a small fragment of glycogen. In the primer initiation, small fragment glycogen act as a primer. When the absence of a glycogen, a protein glycogen act as a primer. Where the hydroxyl groups of amino acid tyrosine act as initial site for binding glucose residue. Initial site for binding glucose residue. Initial binding glucose residue. And then so binding glucose residue. And then the first the glycogen initiator. Produce the first glycogen residue. Then the glycogen itself add as another and the other residues. And when the absence of the residue, and then is glycogen synthesis, and the glycogen is synthesis from alpha 1,4 residue, and glycogen in non-reducing end. In the non-reducing end, the glycogen added glucose. Non-reducing end added glucose with the help of glycogen synthase. The enzyme produce itself, and with the help of UDP convert, and this UDP have the ability to convert, and this UDP are converted back to UDP by nucleodiphosphorkinase. This enzyme help to back UDP into UTP. And third, branching, and naturally the glycogen synthase. This enzyme is the responsible for synthesis of a linear unbranched molecules of a glycogen chain, and And normally the glycogen is a branched chain, which is a branched tree-like structure, and this is responsible for glycosylation 4,6 transfer. This enzyme is responsible for synthesizing glycolysis. This is branching. This enzyme branching in glycolysis, and this enzyme responsible for synthesis branches in 9 to 8 residue, and then branching take place in alpha 1,6 glycosidic bond, and then the enzyme is repeated this process elongation and branching and forming glycolysis.